Hi everyone, I'm Nan Jiang, a postdoc researcher from Dr. Eric Goodford Lab in the Ohio State University, start from 2015. So in our lab, we are most focused on one of the secondary metabolites in plants, and that is the flavonoid. So when you talk about flavonoid, most people is heard about the anthocyanins. The anthocyanin, based on their words, came from the Latin, and it's about the flower and then the pigment. So as you can see, we are interesting is the anthocyanin, how the anthocyanin biosynthesis, and then the regulation of anthocyanin. So the anthocyanin most you can think about is the flowers, which the flower give you the pink color and then the purple color. That's the color. That's the pigment. Is the are the different kinds of the anthocyanins. So I have several projects in the lab. So it's most likely is related to the biosynthesis and then the regulation of the anthocyanin in the Arabidopsis, one of the Dicots model system, and then the mace. So most of the anthocyanin biosynthetic pathway is already been characterized, identified which enzyme, which protein is involved, but the trafficking from the place where the anthocyanin produce, which is ER, and then to the place where the anthocyanin to accumulate all the store, which is the vacuole. So the trafficking precise, how the transportation from ER to the vacuole is still not fully understand. So that's one of the project I'm interesting. So by using the Arabidopsis as a plant system. So what I'm doing right now is by using two ways, the two forward genetics to understand the regulation of the anthocyanin biosynthesis and then the storage. One of the first is by using the EMS mutagenesis to create the mutagenesis lines which can recover. So one of the mutant which is can block the trafficking. So that mutant is already known. So it's cannot accumulate anthocyanin when you do the treatment under the treatment condition. So what I'm doing is I use the EMS mutagenesis to create the mutation and then to do the M2, the next generation screening, to find which the seedling of the Arabidopsis can recover or can restore the anthocyanin accumulation. And then because all the mutant, the background should be the colorless, the seedling should be the colorless. It's like when you culture them under the etiolated, under the dark condition. It's there's no the anthocyanin accumulate, but when there is another, the suppressor is finding it can restore this the mutant, and then that the anthocyanin will accumulate. So I'm trying to find is there other protein or the complex or the multiple proteins will work on the trafficking. So not working not that along not that the known protein. So that is one part. So now I'm identify several lines which is by the different gradient can like partially recover the trafficking, the anthocyanin accumulation. So and the other part is maybe you may know, like the seed coat of the Arabidopsis is most likely the color, the brown color came from the one of the end product of the flavonoid pathway, which is the proanthocyanidins. It's also called the condensed the tannins. So if there is a mutant, it's a block. Is block the flavonoid pathway. So you can clearly see by your naked eye, the seed coat, the color of the Arabidopsis seeds will be the colorless, will be the yellow color instead of the wild type is a brown color. So that project, the second project I'm doing is by using the TDN insertion lines. I try to screening and then to find some the candidate genes, which candidate genes of the lines which is not has not been characterized before, like it might be direct or indirect regulate. It could be the transfer factor or some of the cofactors to regulate the anthocyanin accumulation and the flavonoid the pathway. So I find several lines. I already did the genotyping to find the insertion, and then so now I'm looking for the second allele to confirm, to double check there is like that region is actually, or that region or that gene is actually like the causal reason for the phenotype, the colorless phenotype of the Arabidopsis sigcoat. So the other project is related to the maize. So the maize, as you can see from the McLean-Talker, the transposon, he's using one of the flavonoid, the anthocyanin, the genes as the indicator, the BRONX1 gene, which shows if the transposon is moving, so like it will show the color will change. And then you can clearly see from the kernels. So I'm trying to find more like the fine tuning about those the flavonoid pathway or the anthocyanin pathway synthesis in the maize, to find some like if there is some. I did collect some the RNA-seq the data to see whether the BRONX1, because everyone thought it's a glycosyltransferase, maybe they have some the regulatory the function. It will like the feedback regulation of the whole pathway to either upregulate or downregulate some of the pathway upstream. And then there is some the fine tuning, and then we can also thinking about some the small molecule like the secondary metabolites and the intermediate, the flavonoid intermediate, could also interfere the enzyme function. So that's most likely I'm doing right now. So I know I did a lot the repetitive. So I'm trying to thinking like some the fast way. Okay, thank you. Thanks. Bye.